We are going to look at one of these special tests called thrombophilia screening. And thrombophilia screening has got parameters or sub-tests to measure, and these estimate a person's propensity or chances of forming clots all over the body. And not to confuse thrombophilia screening with hemophilia screening that has to do with bleeding episodes. Now, we are going to have to look at these basic clotting tests. This will lead us into the thrombophilia screening. We've got fibrinogen test and we've got D-dimer test. Both tests are quite related. We will look at the relevance of fibrinogen tests, why they may be low and why they may be high, and their low level, how it's clinically relevant to a person's circumstance. And looking at the fibrinogen test, you will launch the clotting cascade, and as a recap, remember we made emphasis on thrombin. When I circled this thrombin, we discussed thrombin is able to convert factor 13 into its active form, and we said thrombin activates factor 8 and factor 5. And finally, thrombin converts fibrinogen here into its fibrin monomers. That's a clot formation. It's like written a loose fibrin. So if you want to perform a fibrinogen test, the reagent for fibrinogen contains abundance of thrombin, and it is there to convert the present fibrinogen within the patient plasma into fibrin. And this is inversely proportional to seconds. So if you have a very short time to form a fibrin monomer, it means that we've got abundant fibrinogen in there. But the longer time it takes to form a fibrin monomer, it means a very small amount of fibrinogen is present within the patient plasma. So it is estimated from the seconds, and there is a standard graph made with a calibrator that shows the gram per liter amount of fibrinogen based on the time that it takes for thrombin to convert the fibrinogen to fibrin. What are the situations that can cause low fibrinogen level? It can be due to consumption or low production or qualitative problem. So consumption means that the fibrinogen present in the patient's plasma or in the patient's body has been involved in a clot formation in cases like disseminated intravascular coagulopathy or in venous thromboembolism where clot is formed. The fibrinogen genes are three types. It is the FGA, which is FG alpha, then FGB, the FG beta, and then the FG gamma, and these are found on chromosome 4. They are produced as genes, and these genes are translated, and the proteins that comes out of it are assembled within the liver. And therefore, if there is any mutations within the fibrinogen beta gene, which is much involved in the cross-linking feature of the fibrinogen protein in clot formation, it is either going to lead into low production, that is the hypofibrinogenemia, not being produced at all because of the mutation, which is afibrinogenemia, or a dysfunctional type, though much of it are produced, but for its quality it is impaired, and this is called the dysfibrinogenemia. Therefore, the fibrinogen antigen test is an immunoassay test performed with a monoclonal antibody to bind onto the fibrinogen beta chain subgroup, and this beta chain subgroup, if it is in its right structure, will be able to bind to the monoclonal antibody with affinity and avidity, and this assay will estimate how much we've got using the Beer-Lambert's theory with the presence of a substrate that can have a color change. And if this gene has got a mutation, it means that the binding will be very loose or not available, which means that the immunoassay test is going to get a very low results, and this is going to establish the quality of the beta chain subgroup that we have got on these fibrinogen of the patient. Moving on to this D-dimer. Before you can explain properly what D-dimer is, we need to establish clot formation. And that's why I've got all these diagrams available here with all these names written apart. Maybe we'll connect them and make a sense out of it. Now, we understand from our previous discussion that this is factor 12 activated, which is activated by a contact factor. Factor 12 doesn't only undergo the intrinsic pathway of the clot formation, but when it's activated, it also activates its co-contact factors. And we know that prekallikrein and high molecular weight kininogen are all contact factors. So activated factor 12 will convert prekallikrein into its active form called kallikrein. And this pair, factor 12 and kallikrein, will now cleave the high molecular weight kininogen into a stimulus, and this stimulus is called bradykinin. So the conversion of high molecular weight kininogen into its stimulus is performed by a co-conjunction of FOXP12 and kallikrein, and we obtain this bradykinin. Now, what does this bradykinin do? Just as I said, it's a stimulus, and when a stimulus is released, it compares the release of other factors from the vascular endothelium, and this is the vascular endothelium, and this is the subendothelial layers that has got the collagens and smooth muscles and all that. This is tissue plasminogen activator, and this is tissue factor pathway inhibitor. These are present to control coagulation within the blood vessels. Now let's look at this structure. If you look carefully, this is actually a clot that has formed. And when a clot is formed, structurally, it contains these red structures, and these are known as platelets or thrombocytes. And of course, you can see an intertwined multimers and different connections in different directions of these dark strings. And this is what we call the fibrin strands. Now you can see this white structure. It represents plasminogen. It doesn't really form a part of a clot, or it doesn't contribute to clot formation, but it is present in here to bind to the carboxyl-lysine terminal of the fibrin. So it is bound to the fibrin strands here on its lysine terminal. This plasminogen is present to make it easier for the body to have a localized clot dissolution or fibrinolysis in case this clot needs to be dissolved. Now let's get back onto this part so that we can understand the process. We know tissue plasminogen activator has been released because of a stimulus called bradykinin. When this tissue plasminogen activator is released, it goes straight to this plasminogen. Now, this is the plasminogen already forming a part of the clot. When plasminogen is converted into plasmin, this is the active form of plasminogen. So this is a zymogen, which is inactive, and this is the active form called plasmin. Now, the plasmin is able to break down the interwinding strands of fibrin so that this clot can dissolve. And when this clot dissolved, the broken down pieces of this fibrin interconnected strands is called fibrin degradation product. And if you look in here, you can see some of them are still cross-linked and they are larger. The larger ones are called fibrin multimers.
And the smallest part of this cross-linked fibrin degradation product is what we call D-dimer. And mind you, the interwinding structure of fibrin present here are called fibrin polymers and monomers that are insoluble. But when the plasmin, which is an enzyme, acts on these strands, it breaks them down into a soluble fibrin degradation product that are able to be excreted out of the body.
Therefore, the D-dimer is a fibrin degradation product. We get them raised after a clot is formed and it is in a process of dissolution, for instance in venous thromboembolism, in disseminated intravascular coagulopathies. Post major surgeries, they do check for D-dimers, and this gives an idea that the body is dissolving a clot that has been formed previously. And just not to leave this one behind, though we will mention it in our next step, but this is tissue factor pathway inhibitor. It is there released to inhibit the activation of factor 7, which is in the tissue factor pathway, the extrinsic pathway of clot formation.